My name is Ian Denton, and this is mitochondrial ROS production from a paper titled Modeling Mitochondrial ROS Production by the Respiratory Chain. ROS is an abbreviation standing for reactive oxygen species, and these are molecules that are formed through partial reduction reactions of dioxygen. It includes your superoxide radical, your hydrogen peroxide, and your hydroxy radical. Now, ROSs can serve as signaling molecules in a broad range of signaling processes in the cell, and their concentrations are controlled through antioxidant defenses like enzymes such as superoxide dismutase, glutathione peroxidase, and catalase. However, you can have an accumulation of ROS in your system that leads to oxidative stress. Oxidative stress is described as an imbalance between the ROS production and antioxidant defenses. And oxidative stress leads to many different pathologies such as cancer, stroke, neurodegenerative diseases, and age-related diseases. And these arise through your ROS like superoxide radical, hydroxy radical, or hydrogen peroxide, attacking different biological molecules such as DNA, nucleotides, lipids, and proteins. So it's very important to understand where ROSs are being produced and how they can be controlled, monitored, or even stopped. One form of ROS production is your electron transport chain. The electron transport chain is a four-complex system in your mitochondrial matrix that consists of complexes 1, 2, 3, and 4. And there are two ways through it. You can have complex 1 to 3 to 4, or you can have complex 2 to 3 to 4. It just depends on what starting material you are beginning with. And in the electron transport chain, you have the formation of a proton motive force by complex 1, 3, and 4. This is the pumping of hydrogen protons, or just protons, into the inner membrane space. This becomes important later on. Now, out of the four complexes, you namely only see ROS production in complexes 1, complexes 2, and complexes 3. And they account for up to 45% of ROS production in the cell. Beginning with complex 1. Complex 1 is known as NADH ubiquinone oxidoreductase, and it uses NADH in your mitochondrial matrix in a series of reactions to reduce ubiquinone to ubiquinol. The steps reactions include NADH sending its electrons to flavin mononucleotide, and then flavin mononucleotide sending its electrons through iron sulfur centers to ubiquinol one at a time to form the QH2. And during this, you have the simultaneous pumping of four protons out into the inner membrane space through acid-base chemistry where amino acids next to each other are deprotonated or protonated until the proton leaves the system. Now, in complex 1, you have two sites of ROS production, site 1F and site 1Q. Beginning with site 1F, this is your flavin site, so your beginning site where your NADH sends electrons to FMNH. Now, this site isn't really very much supported just because there hasn't been much research into it, but what we do know as of now is that the ROS production produces superoxide radical, as seen right here, and it's namely through the buildup of the semiquinone of flavin mononucleotide. However, one site that we have very strong evidence for is site 1Q, which is the site of ubiquinone reduction by the iron sulfur centers. Now, this occurs namely during reverse electron transport by complex 1, which uses the proton motive force generated by complexes 3 and 4 to bring protons back into the matrix, and due to a high level of ubiquinol to ubiquinone, you end up producing superoxide radical in the mitochondrial matrix due to this. And this is because you end up having a buildup of semiquinone here through the reverse electron transport chain and this interacts with free oxygen to produce your superoxide radical. This is supported by two inhibitors, one being rotenone. Rotenone inhibits complex 1 altogether. So when rotenone was added, you had complex 1 not producing any type of superoxide radical or reactive oxygen species. When rotenone and antiomycin were added, however, you saw that complex 3 started producing reactive oxygen species, but complex 1 was still inhibited. This is also seen right here as well. Moving on, we have complex 2. Complex 2 is known as succinate dehydrogenase, and this enzyme was first introduced back in chapter 16 and as part of the TCA cycle. Now in the electron transport chain, the FADH2 that was formed in this TCA is now used to reduce ubiquinone, similar to complex 1, where your FADH transports electrons through iron sulfur centers to ubiquinone to make ubiquinol. Other sources of FADH2 for complex 2 include outside dehydrogenases, such as your glycerol 3-phosphate dehydrogenase, which it takes glycerol 3-phosphate from the cytosol and reduces FAD with it, and that transports to complex 2 for ubiquinone, as well as you also have acyl-CoA dehydrogenase complex, which is the first enzyme used in the first step of beta oxidation. However, with complex 2, this doesn't form your proton motive force that's being used by either ATP synthase or, as we just mentioned, complex 1 at site 1Q in reverse electron transport. Now, with complex 2, the wild type doesn't really produce any ROS. You see very minimal ROS production. It's only when you have a mutant type of complex 2 that ROS production really takes a hold. And this is at site 2F, which is the flavin site of succinate oxidation, the first reaction of complex 2. There are 2 requirements for this, though. The first of which, there has to be a source of electrons, namely succinate. And the other requirement the site has to be open, namely for the superoxide radical to be produced. Now, this ROS production is at site 2F is supported through both inhibitors as well as concentrations of succinate. So at a specific concentration of succinate, namely 100 to 150 micromolars, succinate complex 2 is very highly involved, or mutant complex 2 is very highly involved in ROS production with the production of superoxide radical resulting in the mitochondrial matrix. However, when you increase the concentration of succinate or decrease the concentration of succinate past this, you don't see any. But you can still have outside dehydrogenases, as we mentioned before, bring in electrons for complex 2, which still results in the production of ROS, species superoxide radical in the mitochondrial matrix. Moving on, we have the first quiz question. In 3 to 5 sentences, I want you guys to explain to me the 2 different sites of ROS production at complex 1 and which and why one of them is heavily supported. Finally, the last complex out of the ETC that functions in ROS production is your complex 3. Complex 3 is known as ubiquinone cytochrome C oxidoreductase, and in this complex, it takes the ubiquinol made from complex 1 or 2 and uses it to reduce cytochrome C through what is known as the Q cycle, which we'll get to later on. But in an overview, the Q cycle has 2 halves where the electrons move from ubiquinol to cytochrome C, and each half pumps out 2 hydrogen protons to make a total of 4 after the cycle has finished. So with with complex 3, it has a very high potential, the highest potential of ROS production, and this is at the Q cycle, namely. So in a perfect Q cycle, what you have is it begins with the first half. Your ubiquinol binds to your QP site, or in this case, your QO site, and the electrons move through one of 2 paths. It either goes through your ISP to your iron sulfur centers to cytochrome C, or the electron goes through cytochrome B low to B high, so finally to a ubiquinone in the QN or your QI site, as here, to make a semiquinone. In the second half, another ubiquinol binds to your QO QP site, then the same 2 paths happen. However, the second path results in an electron being added to the semiquinone made from the first half to form a ubiquinol that then goes and joins the mitochondrial pool. But as stated before, complex 3 has a high potential of ROS production, and there are 3 ways it can make ROS production, namely in the Q cycle. The first of which is known as the early dissociation hypothesis. This is when the electrons from site QP, or your QO in this case, instead of leaving at the same time from ubiquinol, the iron sulfur electron leaves first. This results in a unstable semiquinone being left in the QP site that is able to react with free oxygen, producing a superoxide radical similar to complex 1 and complex 2. The other way is the late dissociation hypothesis. It's essentially the opposite. In this case, your cytochrome B your cytochrome B low electron leaves before the cytochrome C electron from the ISP. This again causes the accumulation of unstable semiquinone at site QP or QO, which reacts with free oxygen to produce superoxide radical. The last of which is kind of different. The electrons move out at the same time, but you can have a short circuit or a bypass result where instead of cytochrome B low going to B high, you have cytochrome B low, like right here, going back to the quinone that was just formed in QP site and adding it back on there, producing another semiquinone, which then reacts with free oxygen to produce oxide radical. Now, compared to complex 1 and 2, complex 3 produces the same ROS, superoxide radical. However, with complex 1 and 2, they produce the superoxide radical in the matrix. With complex 3, you can either produce superoxide radical in the inner membrane space of the mitochondria or the matrix of the mitochondria. Similar to complex 1, the buildup of unstable semiquinone is the cause of your superoxide radical, as in site 3QO, which is the QO site right here, and as well in the 1Q. But it is different than the 1F site, which is caused by the buildup of flavomononucleotide semiquinone. In complex 2 site 2F, this is just due to the site being open and having a specific concentration of succinate. So this leads us to our second question. In about 3 to 5 sentences, explain to me at least 2 ways out of the 3 that ROS production occurs at complex 3, and what ROS is made and where they can be made. So in summary, ROS concentrations are controlled to work as signaling molecules, but you can have an accumulation of ROS that leads to oxidative stress and causes many different diseases. And it is found that the electron transport chain is the cause of up to almost to half of all ROS production, namely being complex 1, 2, and 3. In complex 1, you begin with an NADH and end with ubiquinol, and there are 2 sites, 1F and 1Q. 1Q has a higher maximum capacity for ROS production and is supported by inhibition of birotenone and even the upregulation of production of ROS by complex 3. Complex 2 instead begins with an FADH to reduce ubiquinone, but with wild-type complex 2, there's hardly any ROS production. You will mainly see ROS production in mutant complex 2 at site 2F, where the electrons of FADH2 are instead passed to free oxygen and at a specific concentration of succinate. This is supported by changing the concentration of succinate as well as testing inhibitors for other complexes and other dehydrogenases. Finally, complex 3, as we just recently discussed, takes the ubiquinol from complex 1 and 2 to reduce cytochrome C through the Q cycle. However, ROS production occurs at site 3Q when there's 3 different mistakes in the Q cycle. One being your iron sulfur protein electron leaves first or leaves too late, and then the third one being that a short circuit occurs in the electron in your cytochrome B low, where it goes back to the quinone instead of to the QO site or the QN site. ROS production here supported by the previous inhibitors tested, such as antimycin. This has been mitochondrial ROS production, and I am Ian Denton. Thank you.